Okay, so talking about antibiotic resistance, bacteria that are sensitive to the antibiotic, we describe it as being susceptible to the antibiotic. But during 70 years since the introduction of antibiotic, not so long, 70 years only, okay, most pathogenic bacteria now have already resistant to one or more type of antibiotics, okay? So how they actually gain this resistance when their gene coding of protein protect them from from antibiotics, okay? So for example, penicillin is not effective against tuberculosis because the thick cell wall of the tuberculosis is not very permeable to the penicillin. And the bacterium now, the bacteria now also have the enzyme that break down the penicillin. So penicillin has a structure that can be broken down by the enzyme called beta-lactamase, right? Because of this four-ring member. Four means member, okay? Ring. So other modes, protein that can inactivate antibiotics or they can pump out antibiotics. So now, let us look at this, what actually they can do. Now look at this. Bacterial, okay, prokaryotic cells, they have this the main circular DNA. So not chromosome, guys, it's called circular DNA. They don't have chromosome, okay? Guys, cancel the word chromosome. Always remember bacteria, they don't have a chromosome. Okay? They do have the DNA. They don't have the histone protein also. Okay, so now they have the extra circular DNA known as plasmid, right? Extra one called plasmid. So this plasmid actually very prone to what we call the mutation. Are you clear? And in bacteria, mutation take place. It's unlike in humans that we have or eukaryotic cells, we do have the DNA repair mechanism. For them, no. They don't have DNA repair mechanism. Guess what? Second, we learned about our humans or eukaryotic chromosome. We always come in a pair, diploid cells. So we have a dominant allele and recessive allele. Most of the mutation develop the recessive allele. So recessive allele, the effect will be masked by dominant allele. But for bacterial, they only have one set. So means that whatever mutation take place, straight away the effect can be seen. Get the idea? Okay, straight away the effects can be seen. So what actually happened? For example, in this gene, suddenly because, as I said, that mutation can take place because of the replication. Still remember in the previous chapter? Okay, replication. For example, by right should be G, they put A. For example, by right should be T, they put C. A mutation take place. And this mutation may accidentally, okay, they're lucky enough to form the polypeptide that fall into a protein which can degrade the antibiotic. So means the antibiotic take in, they degrade it. So means that whatever antibiotic enter into the bacterial cells, useless now. Or they may inactivate it. For example, can you see the antibiotic enter? Before this antibiotic can bind to the enzyme, they have the protein that bind to the antibiotic. So this antibiotic now cannot, because the shape totally different already, right? So they cannot bind to the enzyme active site or allosteric site, okay? Or they may have the word called efflux pump. Okay, efflux pump basically means that pump out. Antibiotic enter, they pump out this antibiotic. So the antibiotic have no effects onto the bacteria. Okay. So what actually caused the problem here? It's something like a survival skill, right? Okay, I always say that it's survival skill. Now, for example, okay, if I throw you guys into a jungle, right, a forest, survival skill tell you that you must do something. Okay, going to find food, going to find shelter, avoid the animal, that means avoid a tiger in the jungle, and so on. Survival skill. So bacteria also the same thing. When we use the antibiotic too frequently, whether anything also antibiotic, bacteria want to survive. Bacteria have to do something. Are you clear? And we call this as a selection pressure. Now, basically, if I throw the whole class of you guys into the jungle, those that can survive means that they have the selective advantage. Are you clear? Those that cannot survive is called, they don't have the selective advantage. For example, if I throw you guys in the jungle, you don't have survival skill, so you don't have the selective, you don't have the selective advantage. So bacterial, those that manage to mutate and come out with this antibiotic resistant protein, they have the selective advantage. So antibiotic will act as the selection pressure. Are you clear? If I throw you guys in the jungle, those predator, tiger, those animal inside will become the selection pressure for you guys to survive. Are you clear? So those that can run fast, they can climb up fast, you survive. Are you clear? So bacteria that manage to produce the antibiotic resistant protein, they can survive. So that's why we say that the misuse of the antibiotic now exert a selection pressure on the bacteria. If we don't use the antibiotic so frequently, or we use it correctly, they won't act as selection pressure. Can you see that? Because if you use it correctly, one shot, okay, we finish the entire whole course, 7 days antibiotic, kill all the bacteria. Do they have chance? They don't have chance to mutate. They don't have chance to actually come out with the antibiotic resistant protein. But you take 3 or 4 days, you see that no symptom, no coughing, no sore throat, no fever, and you stop. In fact, you just weaken them. You give them time to mutate and come out with the antibiotic resistant gene. So this antibiotic resistant gene that always take place in the plasmid, okay, is the worry part here, because they can be transmitted. So we have 2 ways for the transmissions, okay? We call it as the horizontal gene transfer or vertical gene transfer. We're going to look at this, how the resistant gene can be transferred from one. You only need one bacteria, you only need one to successfully form this. They always can carry out the gene transfer. Are you clear? So I'm going to talk about the gene transfer later. So I say that the greater the misuse, the greater the selection pressure exerted on bacteria to evolve the resistant. So once they evolve the resistant already, then this resistant gene can be transferred to the next organism. Okay, the next means a bacteria. So antibiotic resistant. So as I said, that the mutation take place, they can have the efflux pump out, they can inactivate, they can have the enzyme to break down. So once they actually, okay, once they already develop the resistance, then this resistant gene can be either horizontally transferred or vertically transferred. So now let us learn what it means by the horizontal transfer and vertical gene transfer. Okay? Before we come back to this diagram again. Okay, so let's look at this gene transfer, either horizontal or vertical gene transfer in bacteria. Gene transfer is a common term, so we have to be more careful with the word we use. We saw the gene transfer in bacteria. Now, what's mean gene transfer in bacteria? Now, let's say we have the first bacteria. So bacteria A, species A. Okay, so species A now have the plasmid. Okay, now due to the mutation, I said that mutation is random and spontaneous. Are you clear? Always take place. So if they manage to come out with, okay, this antibiotic resistant gene, they will survive. Correct? Okay, so let's say they manage to come out with antibiotic resistant gene here. Okay, now what actually happened now? So DNA replication always can take place. When DNA replication take place, so from 1 plasmid, they get 2 plasmid now, or more than that actually. Can you see that? Are you clear? Okay. And Why they carry out DNA replication? They carry out replication because they want to go for the binary fission. Asexual reproduction take place. So when binary fissions take place now, you can see that from one bacterial, you get two bacterial. And they were going to distribute this plasmid equally to two cells. So can you see that? The transfer from one cell to two cells now? The antibiotic resistant, the red color gene from one cell to two cells. So this mode of transfer is known as vertical gene transfer. So vertical gene transfer normally through asexual reproduction, where binary fission takes place. Are you clear? Understand so far? Okay, so vertical. Now, what means horizontal gene transfer? Horizontal gene transfer even more, okay, I don't say even more scary in this case. What I mean here is horizontal gene transfer. This antibiotic resistant gene can transfer between two different species. For example, now you have this, okay? Let me, let's say this bacteria. Species A, right? Just now I said that species A. Okay, I have bacteria species B. So species B also have antibiotic resistant gene, but different, resistant to different antibiotic. Let's say A resistant to penicillin, B resistant to streptomycin, for example. So what will happen here is they also carry out the DNA replication. Okay, both of them carry out DNA replication, so that's why they replicate the plasmid. And through a process called bacterial conjugation, so they establish a channel. So this channel allowed these two to exchange. These two plasmid to be exchanged. So means that now bacteria A, bacteria species B, now you can see that gene transfer take place. The original one, now with a new antibiotic resistant gene now. But they are different species, they're still different species. Can you see that? Are you clear? So gene transfer complete. So this one is called horizontal gene transfer. Through what? Through bacterial conjugation. Okay, through bacterial conjugation. Clear? So horizontal, side by side. So that's why horizontal, vertical go down. Can you see that? So you can take a photo you want it, okay, to know about the gene transfer in bacteria. Okay, so we have the vertical gene transfer versus the horizontal gene transfer. So that's why multiple drug resistance take place. Why? Because you try to imagine this one originally, Okay, the A, you can see the bacteria A originally, okay, only resistant to one antibiotic, but now they're resistant to 2 antibiotics. Are you clear? So if you continue, go for many, many rounds, so it can actually achieve what we call the multiple drug resistant tuberculosis, for example, right? Mycobacterium tuberculosis. Okay? Okay, so for activity 13.15, antibiotic resistant, okay, so we have the horizontal gene transfer. So it can be by conjugations, or sometimes virus can help to take in, okay, so by transductions, viral transductions, and also sometimes you can see that some resistant gene, naked ones can be taken in, okay, because of the cell slides. Or this is called horizontal gene transfer. Vertical gene transfer, you can see that non-resistant. If one of them managed to get it, then you can see that they start to multiply because they won't be able to kill by the antibiotic anymore. So give them chance to actually multiply, and you can see that we only need one of them, okay? And bacterial multiplication is very, very fast, roughly about 20 minutes to 1 hour, one round. You can always calculate it from 1 to 2 to 4, 4 to 8. If they say 1 hour, they replicate one time. After 24 hours, it will be 2 to the power 24. Can I see that? Are you clear? So means that your number will be very, very high. Can we kill them by using an antibiotic? No, we have to choose the correct antibiotic. If no, you won't be able to kill them. Are you clear? Because antibiotic resistance. Okay. So complete this 5 minutes. So let's look at the answer now. So antibiotic resistance, it can arise when existing gene within the bacterial genome changes spontaneously. I say that spontaneous mutation always take place, give rise to nucleotide sequence that code for slightly different protein that is not affected by the antibiotic. So this chain DNA is known as a mutation, or gene mutation. So when someone takes penicillin to treat bacterial infection, bacteria susceptible penicillin will die. If the dose is not followed, perhaps because people stop taking penicillin when they feel better, okay, so some of these susceptible bacteria survive, and if any mutation occur, this might confer resistance. So next time there is an infection with the same strains of bacteria, penicillin may not be effective anymore. So bacteria have only one copy of each gene. So since they have a single loop of double-stranded DNA, this means that the mutant gene will have immediate effects on any bacteria possessing it. So these individuals have a tremendous advantage. So bacteria without this mutant gene will be killed, while those bacteria resistant to penicillin survive and they reproduce. When they reproduce, they will going to pass the gene to the next generation. So they can reproduce asexually by binary fissions. Okay, and the DNA of the bacterial fibroblasts is replicated and the cell divided two, and each daughter cell is now receiving a copy of DNA. So it's very, very rapid in the ideal condition, the process. And even if there's initially only one resistant bacterium, it produces 10,000 million descendants within 24 hours. A large population of penicillin resistant strain of bacteria would result. And this method of spreading is known as vertical transmission or vertical gene transfer. Okay. So the genes for antibiotic resistant often occur on the plasmids with a small loops of double-stranded DNA. So plasmids are quite frequently transferred from one bacterium to another, even between the different species. So this happens during conjugations, and this mode of the transfer is known as horizontal transmission or horizontal gene transfer. Okay, it can be occur when one species of bacterium can be passed on to another through the transduction process, the process with which bacterial DNA is moved from one bacterium to another through a virus, for example, bacteriophage or phage. Okay, and bacteria also have the ability to acquire antibiotic resistant gene from their environment. Okay. Now, we do have this, what we call the last resort, okay, of the antibiotic called vancomycins, but now vancomycin also can be used because Enterococcus faecalis and Staphylococcus aureus also resistant to vancomycin. No, it's not last resort anymore, okay? Recently, antibiotic named carbapenems also, we want to use the last resort, but also can see that we have the pneumonia, okay, caused by this Klebsiella pneumoniae, also that means resistant to this antibiotic anymore. In 2017, you can see that the WHO published the first ever list of antibiotic resistant priority pathogen. So a lot of them, they are gram-negative. So what is gram-negative? If you look at the bacterial, gram-negative and gram-positive, gram actually is a stain. Okay, it's a stain, it's not the weight, okay, the gram. So gram-positive means that you can see that they will appear as the purplish color, but gram-negative, they appear as the pinkish color. Are you clear? And if you look at gram-positive, you look at the peptidoglycans, there are multiple layers of peptidoglycan. And if you have the antibiotic that target peptidoglycan, you can see that immediately can actually target the cell wall. But if you want to target the peptidoglycan here, no, because they do have this outer membrane. For gram-negative, they double membrane. So most of the antibiotics actually are not effective towards the gram-negative bacteria. Are you clear? Okay. So you can counter stain with the red color. Now, this is for example, for your for your information only. Do not need to memorize this, because in the exam, if a gram-positive, gram-negative bacteria come out in this question, come out before, okay, so you just need to look at the diagram and answer the question. Are you clear? You're not required to memorize. Okay, so for information only. So example for gram-negative, we have the Pseudomonas aeruginosa, gram-positive Bacillus anthracis. Bacillus anthracis causes the anthrax, the disease. Gram-negative reactions can be decolorized to accept counter stain, safranin. So therefore, give us the red or pinkish color. Okay, it's a gram-negative. So bacteria stain red or pinkish because they cannot retain the gram stains. Okay, the purplish color. Bacillus, they retain the crystal violet dye and stain dark purple or purple. Okay. Thickness of the cell wall, you can see that roughly about 70 to 120 angstrom. Okay. But the cell wall for the gram-positive, 100 to 120, so thicker in this case. Number of layer membrane, 2 layers, gram-positive 1 layer. Peptidoglycan, thin, basically gram-negative, only single layer, but thick for multiple layers. Cell wall composition, they have high lipid content, low murine, but this one low lipids but high murine. Are you clear? Now what's high murine? Can you see that? High murine, low lipid, only 1 layer. This one, 2 layers, but less. Are you clear? So high lipid, low murine. High murine, low lipid. Okay. Outer membranes present. Liposaccharides content, LPS. Can you see that? LPS present in the gram-negative. And antibiotic resistant, more resistant to antibiotic compared to the gram-positive. Okay. So again, right now, guys, for this, for this part, guys, just for information only. If the question come out, they will give you the diagram. You do not need to memorize. Just look at the diagram, then you differentiate which one is a gram-positive, which one is a gram-negative. Okay. So antibiotic resistance is putting the achievement of modern medicines at risk, because organ transplant, chemotherapy, surgery, those can cause, infections. So you want to prevent infection, we use antibiotics. But the thing here is antibiotic now resistance. Okay. Fortunately, a bacterial resistant, a particular antibiotic may not resist to other antibiotics. So we can do the semi-synthetic antibiotic when you change the chemical structure. So we do have some new antibiotic developments, but none of them is expected to be effective against the most dangerous form of antibiotic-resistant bacteria, according to WHO in 2016. It means that the speed of the bacteria to develop resistance is faster than with our discovery. Do you get what I mean? Okay. So well, there are some new antibiotic development, but none of them are effective. And in fact, we do have some other improvement and developments. Okay, they pump in the money, but so far not really successful. So the new antibiotic treatments nowadays in the Dutch biotech company, they use a virus. So because some bacteria can be infected by virus, so we can use the virus to kill them. It's something like biological control. Are you clear? Okay, we use something like biological control, but in this case, we use the virus just for information. And this one, Lam Sujie, I think that's quite famous, this girl. Okay, because why? Because she was a Malaysian, but studied in Australia. So she actually published a paper saying that by using a peptide polymer, they can rip apart the cell wall. Can you see that? So this is a bacterial. So by using treatments, okay, one-time treatments, can you see that they tag with the green color? So this actually can help us to rip apart. So it means that they can program the polymer to rip apart. So it's not antibiotic. Can you see that? It's something like mechanical. So the polymer that bind to the cell wall and then rip apart the cell wall. So when you break the cell wall, you can kill the bacteria. So this is non-antibiotic approach to kill the bacteria. Are you clear? Okay. So what are the strategies to minimize the antibiotic resistance? So again, we use antibiotic only when appropriate and necessary. Do not prescribe them for viral infection unless the viral infection caused the secondary infection by bacteria. Okay. Reducing the number of countries in which antibiotics, okay, are sold without a doctor prescription, and avoiding the use of wide spectrum antibiotic and use instead an antibiotic specific to the infections. Okay. Antibiotic sensitivity test is conducted. Later, I will show you what's mean antibiotic sensitivity test. Make sure the patient complete the course of medication, making sure the patient do not keep unused antibiotic for self-medication in the future. Changing the time antibiotic prescribed for certain disease so that the same antibiotic is not always prescribed for the same disease. And avoid using antibiotic defarming to prevent rather than to cure the infections. Are you clear? Okay, so those are the strategies. So you can take a photo of those online. Please take the photo. Okay, done. Okay, so now next. So what's mean antibiotic sensitivity test? Antibiotic sensitivity testing or antibiotic susceptibility testing is usually carried out to determine which antibiotic will be most successful in treating the bacterial infection. So we can use the diffusion method or dilution method. So in our syllabus, we're going to learn about this diffusion method. So what we're going to do here is we have the agar plate. So this agar plate, actually, we culture them with the bacteria. Let's say sample bacteria from a infection wound. Are you clear? Infected wound. So we culture them. So these originally, they are full with bacteria. Then we use the These actually contain the different type of antibiotic. Are you clear? Antibiotic A, B, C, D, E, F, G, H, I, J, K, L, for example. Are you clear? And then we place on the bacteria. And because of the diffusion, so this antibiotic diffuse out, so antibiotic that diffuse out kill the bacteria. So it creates what we call the clear zone. Can you see that? The clear zone means that without the bacteria. Why? Because bacteria cannot grow here. Bacteria are killed. Are you clear? But you can see some of them no zone at all. Basically means that they are not effective in killing the bacteria. So from here, you don't choose this. No. This one, no, not useful, not useful. Maybe this one, okay. So then you prescribe this antibiotic for your patient. Are you clear? Okay, so means that we use the one. So this one may be resistant. This one be small. Okay, so we use those antibiotic that give us a more larger clear zone. Okay. Okay, so because of the convenience, efficiency, and cost, this diffusion method is probably the most widely used method for determinations of the antibiotic resistance. So a grow medium usually Mueller-Hinton agar first evenly seeded throughout the plate. Okay, it isolates. So with the isolates of interest that have been diluted at a standard concentration. Okay, don't worry about this. Then we commercially prepare this, okay, with particular antibiotic. So the test antibiotic immediately begin to diffuse toward the yeast, creating a gradient of antibiotic concentration. So in the agar, such that the highest concentration is found close to the yeast and the decreased concentration further away from the yeast. So after an overnight incubation, the bacterial growth around each dish is observed. If the test isolate is susceptible, means that it's sensitive to a particular antibiotic, a clear area with no growth will be observed. So this zone is known as zone of inhibition. So diameter of the zone can be measured in mm and compared to the standard interpretation charts used to categorize the isolate as susceptible, intermediately susceptible, or resistant. So MIC measure cannot be determined from the test, which simply classify the isolate as susceptible, intermediate, or resistant. So if let's say you measure, oh, this diameter is too small, maybe resistant. Are you clear? Oh, this diameter is large, so susceptible. This is how it's classification. Okay. So the last part, we look at this methicillin-resistant Staphylococcus aureus called MRSA. Oh yeah, MRSA. So this methicillin-resistant Staphylococcus aureus is a gram-positive bacterium, genetically different from other strains of the Staphylococcus aureus. Now, coccus means round shape. Staphylo means forming a bunch, a grape. So the bacteria is something like this. Can you see that? It's a coccus. This form is called Staphylococcus. We have another one called Streptococcus. Strepto means form a chain. Are you clear? Streptococcus. Okay, so this is Staphylococcus. So MRSA is any strain of Staphylococcus that developed through horizontal gene transfer and natural selection, multiple drug resistant to beta-lactams antibiotics. So means that, so some of these antibiotics, like methicillin, oxacillin, penicillins, okay, and cephalosporins, those kind of antibiotics have no effect onto this bacteria, basically. Are you clear? Okay. So strain unable to resist these antibiotics are classified methicillin susceptible Staphylococcus aureus called MRSA, not MSSA. Is it unable to resist? Okay, unable to resist is called susceptible, correct? MSSA. MRSA means resistant. So MRSA often means are responsible for several difficult to treat infections in human. For example, people with open wound, invasive device, and weakened immune system. So it's difficult, and you can see that's the effect here. You can see that the lesions. Okay, those are the wound caused by MRSA. This one is the most, how to say that, non-severe case. Severe one, you can see that because this MRSA, this bacteria basically is a flesh-eating bacteria. So when infection takes place, like for example, at your muscle, one of the way, because we cannot stop them, you have to remove the entire muscle. Surgery. Are you clear? And it's not guaranteed. It can spread to many parts of your body. It's called organ failure and it kills. So MRSA until now, it still can't be treated. Why? Because antibiotic is impossible to cure them. Surgery is one way. Are you clear? Okay, we can use a very strong antibiotic, but again, when you increase the antibiotic to a higher dose, it may cause the organ failure as well. So the doctor facing a difficulty to balance between these two. Too high dose of antibiotic, kill patient. Too low, cannot kill the bacteria. So this is what actually happened to this MRSA or MRSA. Okay, so with this, I've officially done the AS syllabus. Okay,